Good evening, everyone, and welcome to our final lecture for Living Systems 1. This is lecture 18. We'll be looking at the second part of this lecture on classification. So we've looked at our prokaryotes, bacteria, archaea. We looked at kingdom fungi and the protists. And we even looked very briefly at viruses. Okay, tonight we visit Kingdom Plantae. So we'll start with the very earliest plants, which were thought to have colonised land about 410 million years ago. And these early plants were thought to have been very small and mostly confined to wet margins of wetlands and rivers. And of course, that was before they evolved all the structures that were required to enable them to inhabit more arid inland areas. The closest relatives to today's plants are green algae, or certain groups of green algae. One group is called the chlorophytes, and plants and chlorophytes both share certain characteristics. In particular, they've done a lot of DNA comparisons between the two, and they certainly share a lot of DNA sequences in their genes. They also have the same two major chlorophyll pigments called chlorophyll A and chlorophyll B. Their chloroplasts have very similar structure. They both have cellulose in their cell walls and both use starch as their storage material. So the transition from water to land required certain structural adaptations, mostly in the area of maintaining water balance, but also to enable plants to extract water and nutrients from the soil. They needed to develop ways of transporting water and nutrients around the plant. And of course, this would enable plants to become larger over time, and also a means of supporting the plant, also enabling plants to grow much larger. So let's look at the specialised features that plants developed over time. Firstly, they had to develop a cuticle. And you remember from a previous layer that this is just a previous lecture that this is just a waterproof layer, a waxy layer that covers the above ground parts of plants. So it's not a layer of cells, it's just a waxy layer that's actually secreted by those outer epidermal cells of plants. Plants also evolved to produce this tough substance called sporopollenin, and this is a substance that protects both spores, that is, spores are produced by certain groups of plants, and also pollen. Plants also evolved these structures called stomata, and we've also covered this briefly in a previous lecture. And stomata are these tiny pores on the surface of plant leaves and stems that allow carbon dioxide to diffuse into the plant. And of course, carbon dioxide is critical for photosynthesis. Plants evolved a vascular system. Well, many groups of plants evolved a vascular system. You remember that this is a transport system within the plant that allows the transport of water and nutrients through part of the vascular system that's called the xylem, and movement of manufactured carbohydrates via photosynthesis. And the part of the vascular system that transports these is referred to as the phloem. Plants also developed this substance called lignin. And you know that lignin is a very tough phenolic substance found predominantly, but not only, in vessel element walls in the xylem, and this prevents collapse of xylem elements under the pressure that they're exposed to. Okay, the very early plants were thought to consist only of stems. But later on, plants developed roots and leaves, and this facilitated this division of labour amongst specialised tissues and organs. All right, now let's look at the major, or I think we've covered all the different phyla found within the kingdom Plantae. And we'll start with this group. These are the non-vascular plants, meaning that they never evolved that vascular system which contains xylem and phloem. 3 phyla here: phylum Hepatophyta, liverworts, the hornworts, and the mosses. And you don't need to remember these names. This is just for your interest, and they're pictured here on this slide. So the non-vascular plants obviously lack a vascular system, and they also lack lignin. So you can infer from these 2 points here that these plants tend to be very small, and that's exactly correct. Okay, our next group of plants are the spore-producing vascular plants. So these plants do contain a vascular system, but they produce spores rather than producing seeds, so distinct from seed-producing plants. 4 phyla within this group, including club mosses, Xylotum, the horsetails, and the ferns. So vascular system is present. They do contain lignin also. And a couple of examples there for you. Now finally, our very important group, the seed-producing plants. So this is divided into 2 groups, the gymnosperms and the angiosperms. So the gymnosperms are non-flowering seed plants. So rather than producing seeds within structures called flowers, they produce their seeds within cones. 4 phyla listed here, including quite a successful group, the conifers, which everyone's familiar with. Then we have the cycads, and we have a cycad pictured here with one of its cones, ginkgos, and the neophytes. Then we have the angiosperms. These are our flowering seed-producing plants, and the most important group for us are the angiosperms. So 1 phylum within this group, Magnoliophyta, which are all the flowering plants, and obviously very, very successful group with well over 250,000 species. These ones are our major producers in terrestrial ecosystems, providing food, timber, fibres, spices, and medicinal drugs that sustain the entire human population. You'll also remember from a previous lecture that we looked at the concept of secondary growth, and it's this type of growth that allows plants to become very large. And you might recall we talked about the vascular cambium, so this additional region of meristematic cells, and the vascular cambium gives rise to secondary xylem and secondary phloem, and the secondary xylem adds girth to the stem. So vascular cambium generates new secondary phloem and new secondary xylem. So the phloem towards the outside of the cambium, xylem towards the inside. All right, the angiosperms, our flowering plants, are further divided into 2 groups. Class Liliopsida, which are the monocotyledons, or monocots for short, and class Magnoliopsida, otherwise referred to as the dicotyledons or simply dicots. And you might remember we mentioned the dicots also in a previous lecture. So let's look at some of the characteristics for each of these 2 groups. Monocots, the seeds contain 1 cotyledon, otherwise referred to as the seed leaf. So really we're referring to the embryo within the seed, and that embryo develops one seed leaf or one cotyledon. The flower parts are found in threes or multiples of threes. The leaves have parallel veins. The vascular bundles are scattered randomly in the stem. So that's the monocots. The dicots, the embryos within the seeds have two cotyledons. Flower parts are found in fours or fives or multiples. Leaves have netted or branched or reticulate venation, and the vascular bundles form a ring within the stem. So we can look at those characteristics on this slide. So we've got the monocots across the top and characteristics of the dicots at the bottom there. So let's look at the monocots. So here we have a monocot seed, and this region here represents the developing embryo, and part of that embryo will actually form one cotyledon. And that's what you see when these seeds germinate, the emergence of one cotyledon. In contrast, the dicots, the seed isn't shown here. Here we can see the germinated dicot seed and the first two leaves that appear, and they're not considered to be true leaves. These are the two cotyledons which developed as part of the embryo in the seed. Monocots, here we have a cross-section of a stem, and you can see these little dark spots. They represent the vascular bundles just scattered randomly throughout the stem. Whereas a cross-section of a dicot stem reveals vascular bundles in a neat ring within the stem. Leaves of monocots tend to have parallel veins, and you might recall that the veins represent vascular bundles that you can actually see when you look at the leaf. So parallel veins in the leaves, whereas dicot leaves have a branching or a reticulate venation. All right, monocot flowers. The flower parts are present in threes or multiples of threes. This says flower petals, but it actually refers to all the flower parts. The petals, the sepals, the stamens, and so on. Whereas dicots, flower parts, are found in multiples of 4 or 5. Let's look very briefly at the reproductive cycle of the flowering plants. Very briefly, and we'll look at this in a lot more detail in the second semester. So if we take just a couple of structures from this flower, so this central structure here, which is the carpel, and that's pictured here. That's the female reproductive organ. And then one of these little stalks here, that's the male reproductive part of the flower, and here it is here, enlarged, the anther. So within the female carpel, you can see this region here is referred to as the ovary. The ovary contains one or more ovules, and inside the ovules are one or more egg cells. Each egg cell is fertilised by a sperm cell, and that sperm cell arises from the pollen that's produced in the male anther. So once the egg cell is fertilised, now we'll just jump over here, it starts to develop into an embryo, which you can see here in green. And then a seed coat will form, a protective seed coat will start forming around that embryo at the same time as the embryo is developing. And then this outer layer here in brown, this was originally part of the ovary wall. And that part of the ovary wall starts to develop into a fruit. And that fruit surrounds the seed coat, the developing seed coat. So that fruit could be, for example, the fleshy part of a grape berry, or the fleshy part of an apple, for example. So let's just briefly look at fruits. So fruits protect the seeds and aid in their dispersal by fruit-eating animals. There are different classifications of fruits. So simple fruits are derived from an ovary of a single flower with one or more carpels, for example, grapes, apples and peas. Aggregate fruits that are derived from clusters of carpels in a single flower, for example, raspberries and blackberries, and multiple fruits derived from clusters of carpels from several flowers, for example, pineapples. Okay, moving on now, kingdom Animalia. So let's start with how do we define an animal? Just some of the characteristics which are used to define animals are listed here. Animals are all multicellular. There is a division of labour with specialised cells, and in all animals other than the simplest, the sponges, those cells will form tissues and organs. All animals exhibit embryonic development from a single cell called a zygote, and all animals are heterotrophs. All right, so under that kingdom level of kingdom Animalia, we have two subkingdoms. Subkingdom Parazoa, which includes those very simple sponges, and subkingdom Eumetazoa, all other animals. Within the Eumetazoa, the animals are divided into two groups, the radially symmetrical animals and those animals that exhibit bilateral symmetry. Is everyone familiar with those terms, or should I go over them quickly?
If you don't mind, Silvana.
Go over them? Sure. All right. So radial symmetry. So if you had an animal that looked like that, then you could divide that animal into two halves using any plane, and you would still get two similar halves. So that's referred to as radial symmetry. But when it comes to, that's radial symmetry, bilateral symmetry, and here comes one of my special drawings. Okay. And that's even worse than what I have previously drawn. Okay, what sort of animal do you think I'm trying to draw here?
Oh, like a lizard?
Platypus. That's a yes. I'll take that. I'll take platypus. Most students guess lizard. I never really know what I'm drawing. I'm just trying to draw something with legs and a tail and a head. But yes, thank you. That is actually a platypus. Thank you very much. All right, bilateral symmetry means that you can only divide using one plane, which I'm trying to draw a straight plane that way, to give two halves, two similar halves. So that's referred to as bilateral symmetry. Okay, does that make sense?
Mm-hmm. Yeah, thank you.
Yeah. Okay, so radially symmetrical animals and bilaterally symmetrical animals, which are placed into the bilateria. The bilateria is further divided into two groups, the protostomes and the deuterostomes. The protostomes include the flatworms, segmented worms, mollusks and arthropods. The deuterostomes, the echinoderms and the chordates. So let's look at those two terms, protostomes, and that takes us back to very early on in embryo development. This little pocket forms, this little opening forms. So this embryo consists of multiple cells, undifferentiated cells, and the first little pocket or opening that forms will actually develop eventually into the mouth. Well, in those animals where that opening develops eventually into the mouth of the animal, they're referred to as protostomes. Now, deuterostomes, on the other hand, I'm trying to draw the same thing. The first opening that develops in the deuterostomes, that opening will develop into the anus of the animal. And the mouth will develop at this end. In the protostomes, the anus will develop at the other end of the embryo. So that's all that means, those two terms, protostomes and deuterostomes. Okay, here we have a phylogenetic. Yes.
Oh no, it was a slightly facetious comment for the end of semester, I guess. That I know some people where that might have got mixed up with the first opening.
They might have got that mixed up. Did you say?
Yeah. Well, I think some people might talk necessarily from their other end.
Oh, very good. I don't know who you're referring to there, Liz, but I'm sure you're right. Yes, good way to finish semester, isn't it? All right, so here we just have a phylogenetic tree of the animal kingdom. And let's have a look down here. So here we're looking at the eumetazoans, and we can see that that's splitting off here to the left. So animals with radial symmetry, which includes just these two branches here. And then to the right, we go to the right, bilateral symmetry. So all these other branches here are animals that exhibit bilateral symmetry. Now within the bilaterians, there's another split here into the protostomes and the deuterostomes. Okay, so you can see roughly how that works. All right, we're going to visit some of the phyla within Kingdom Animalia, just very briefly. So the first one, our very, very simple animals, the sponges. And of course, we all know sponges are aquatic animals, and they have simple cell layers that are not organised into specialised tissues or organs, but they qualify as animals because they have these particular cell layers that characterise all animals during the development at the embryo stage. Something else that we'll talk about in a little bit more detail in second semester. Our next group, the polyps and medusae, otherwise known as the cnidarians because they belong to phylum Cnidaria. These are animals with radial symmetry and they have two tissue layers in the body wall. So this includes jellyfish, corals and sea anemones. Does anyone recognise this one here?
Mm-hmm.
Very, very Australian.
Portuguese man or is that what they call it?
Could be. Blue bottle, yes. Yeah, blue bottle. Yeah. Yeah. All right, our next group are the flatworms belonging to phylum Platyhelminthes. This group includes both free-living and parasitic flatworms. This group includes flatworms, flukes and tapeworms. And they call them flatworms because they have this, where's the pen? Yeah. If you were to cut one in half and then look at a cross-section of it, they have this appearance. So they have that flattened appearance, dorsoventral flattening, rather than a circular cross-section. All right, just an example shown here. This is the liver fluke, which has two hosts. Here we have the primary host, in this case it's a sheep, and there's also a secondary host. So the liver fluke completes its life cycle in two different animal hosts. So the adult fluke will lay eggs, which come out in the feces of the animal. The egg hatches into the first stage, larval stage. the larvae actually penetrate the flesh of only specific species of snails. And there it goes through several larval stages, eventually exits, burrows out of the flesh of the snail, killing the snail in the process, and then eventually the larvae will form this protective layer that is, they're said to encysst, and they do this on grass and other herbage where they're ingested once again by stock, and the life cycle starts again. Actually, a bigger problem in Australia than what was thought a few years ago. They've been doing a lot of studies on the prevalence of liver fluke at various properties across Australia, and it's actually quite a big problem. Oh, all right, our next group are the segmented worms, phylum Annelida, and everyone's familiar with this lovely earthworm, and of course you can very easily see all those segments that make up the body. Hence the term segmented worms. This includes marine worms, earthworms, and leeches. They exhibit the primary characteristic of metameric segmentation, and that means that there's a repetition of certain functional units within each of those segments. So there might be a repeated cluster of nerve cells or repeated clusters of reproductive cells in each segment. Next group are the mollusks, phylum Mollusca, and this includes clams, mussels, snails, slugs, octopus, and squid. Here are the characteristics of the mollusks, but not every mollusk will have every one of these characteristics. So we can look at these in this image of a generalised mollusk. So let's start with this part here, which is referred to as the visceral mass, and this contains all the organs. Then covering that is this brown region called the mantle, and the mantle in some of the mollusks will secrete a shell. Then we have here a single ventral foot, obviously for motility. And then within the mouth, there's structure like a modified tongue called the radula, and the radula consists of these rows of rasping teeth. So that's the mollusks. Next we have phylum Nematoda, and these are the roundworms. They may be free-living or endoparasites. And here's an example of a disease of the lymphatic system that can be caused by one of these nematodes. 25,000 species, and many of them are parasites of plants and animals. Next, probably the most diverse of the phyla, phylum Arthropoda. Spiders, crabs, millipedes, crabs again, yabbies, and insects. Characteristics of the arthropods includes jointed limbs, an exoskeleton that contains chitin, metameric segmentation, but with a difference. This type of metameric segmentation is associated with what's called tagmatization. That is, several segments of those segments will be clustered together into functional regions or tagmata. So the functional regions might include a head, a distinct head, a distinct thorax, and a distinct abdomen. So that's metameric segmentation with a bit of a difference. Next phylum is Echinodermata, which includes sea stars, sea cucumbers, sea urchins, and so on. Next, phylum Chordata, acorn worms, sea squirts, lancelets, and vertebrates. These all exhibit bilateral symmetry. The major characteristics of the chordates are shown here. 1, the presence of a notochord at some stage, pharyngeal slits, and a dorsal hollow nerve cord. Okay, so the presence of these three characteristics explains why The vertebrates, which everyone's familiar with, are classified in the same group as something seemingly very unrelated as a worm.
What's a notochord?
Oh, I'll explain that in just a minute.
Okay.
Yeah. All right, so we're going to focus in a minute just on the vertebrates, but let's look at those 3 characteristics. All right, here they are again. And these 3 characteristics are present at least some stage of the animal's life cycle. So the notochord is a dorsal rod, which is situated between the nerve cord. So of course, so think about your spinal nerve cord and the gut. So it's found in between those two, and it forms an attachment point for blocks of muscles. Some animals retain this notochord as an adult. Other animals lose the notochord, and I'll come back to that also in just a minute. The presence of pharyngeal slits at some stage in development. So these are paired openings in the pharynx, and they evolve into quite different structures. So for example, if we think about pharyngeal slits in fish, they evolve into gills, exactly right. They evolve into gills that are used for gas exchange, but pharyngeal slits in humans actually develop into something very different. Any idea? Ever heard of what our embryonic pharyngeal slits actually develop into? No? That's all right. We'll come back to that in just a second. And next we have the dorsal nerve cord. So of course the spinal cord. This is a hollow nerve cord which is situated above the notochord, and in some animals it expands at one end to form a brain. All right, so let's look at those 3 here. So this is a human embryo that we're looking at, and we can see those 3 characteristics here. So in blue, we have the dorsal nerve cord. So of course that's our spinal cord, and in humans it expands here to form the brain. So that's straightforward. Just below that, so this brown region here, this is the notochord, what's called the notochord. Some animals will retain this, we don't. This will actually develop into the vertebrae, the spinal vertebrae, which actually replaces this notochord. And then here we have in green the pharyngeal slits, which, as we said earlier, in fish these will develop into the gills, but in humans these actually develop into parts of the inner ear. So you might have heard of the eustachian tubes. Certainly if you ever have sinus problems, you'll know all about eustachian tubes. But these slits also develop into the tonsils, certain glands, and parts of the jaw also. So very interesting that you can have a very, very young embryo from a human or from a fish or from other animals, and they look very, very similar. At the embryonic stage, it's very difficult to actually tell them apart. But it's a very complex movement and gradients of proteins that are set up within the very young embryo that determine what that embryo, what final form that embryo will take. And of course, we won't go into those details here. All right, subphylum Craniata includes jawless fish and vertebrates. Characteristics of the craniates: they have a head with a skull or cranium, and they have a brain. Here we have the jawless fish, which includes the lampreys and hagfish. Characteristics: they have a cartilaginous skeleton. The notochord persists in this group of animals, unlike humans. And the jawless fish, of course, lack jaws. Okay, on to the vertebrates, which includes fish, amphibians, birds, reptiles, and mammals. We'll be looking just at the mammals. Apologies to aquaculture students, but you'll get lots of taxonomy and systematics from Sadiq. Okay, characteristics of the vertebrates. Spinal vertebrae will replace the notochord in the adult. Projections from the vertebrae will protect the nerve cord and aorta. Specialised cells, which are very characteristic of the vertebrates, called neural crest cells. These are cells that will form during the development of the embryo, and they're very unique in that they actually detach from where they're formed. They detach, they differentiate, and they start moving through the actual embryo. And then when they reach their final location within the embryo, they give rise to clusters of different nerve cells, different glands, and so on. So this is something that's very unique to the animals, these neural crest cells. And another characteristic is the formation of dentine and enamel to form teeth or denticles. It sounds very odd for birds to have teeth or denticles, but they do, as young embryos, they do have little teeth buds that never develop into teeth, but they have the starting of those teeth. So I suppose that's why they fit this group. All right, let's now just look at the mammals very briefly to finish off. Mammalia, characteristics. There are actually quite a few different characteristics of mammals, only two are shown here. Presence of epidermal hair at some stage of development, and milk production from mammary glands. Two subclasses within class Mammalia, subclass Prototheria, which includes the order Monotremata, the monotremes, the egg-laying mammals. And then we have subclass Theria, which includes the marsupials and the placental mammals. So we'll visit them very briefly. Here we have our monotremes, the only egg-laying mammals. This group is represented by only 3 species, the platypus, which I drew very nicely before. Thank you once again, Liz. And 2 species of echidna. And the monotremes represent the most ancient lineage of mammals. The metatherians, these are the marsupials, and this is the dominant group of mammals in Australia. 272 species, mostly in Australia. This includes quolls, bilbies, opossums, koalas, kangaroos, and bandicoots. The marsupials are characterised by birth of their young at a stage that's equivalent to the fetal stage in the eutherians or the placental mammals, which we'll look at next. So this is illustrated here. So we can see this very young kangaroo that's been born, and now it's finding its way to the pouch, the marsupium of the mother kangaroo. So you can see that it's barely more than a fetus. So they're born at a very young age. So the young find their way to the pouch, the marsupium. Now the female marsupials have a left and right uterus and 2 vaginal canals. The males have a penis that is bifurcate or forked, which you can see in this image here. So some odd characteristics for the marsupials. Okay, and finally we have the eutherians, the placental mammals. A very successful group with over 4,000 species, and the placental mammals are the dominant mammals found in all countries apart from Australia. Thought to be the most recently evolved, about 75 to 45 million years ago. Characteristics of the placental mammals. They do not lay eggs. That's termed viviparous. They have a very specialised placenta called the chorioallantoid placenta, which basically means that the placenta is derived partly from the embryo itself and partly from the wall of the uterus. So this is very different to the placenta of the previous group, the marsupials. Placental mammals, the females have only a single vaginal canal and a single uterus, and in males the penis is not bifurcated. And we finish living systems with a phylogenetic tree of the eutherian mammals. And that's it. Congratulations. You have now completed Living Systems 1. And I hear silence. Oh, well done, everyone. We've come a long way.
Yes, it's certainly picked up a lot in the last few weeks, I think, like in terms of the complexity of the...
Oh, it certainly has, yes. It always does.
Yep.
So we like to start slow the first few weeks and then start building on that. Now, you've done very well to get through. And now let's just briefly look at your last set of tutorial questions, which you've probably answered most of these already. So don't forget to check your answers via the non-assessed online quiz. And now let's look at the exam. The all-important final exam. Well, maybe not all-important, because you've probably worked out your marks so far. So if you can add to that your prac report marks, then you can already get a rough idea of what you're sitting on at the moment. All right, so let's look at the final exam for Living Systems. It's the first exam in the exam period, Monday, 4th of June, starting at 9 o'clock with 15 minutes of reading time. And then after that, we have 90 minutes of writing time. So the exam will finish at 10:45 a.m. All right, let's talk about reading time. So you've got 15 minutes in which you should not touch any of your pens or pencils. If you do, the invigilators will get very upset. All right, so just keep that in mind. And often you're just busy concentrating on reading the exam and you happen just to pick up a pen. Don't be surprised if the invigilator is straight over to you saying, put that down. Okay, 15 minutes reading time. What do you do during that time? Obviously, you read through the exam or skim through the exam. That will enable you to pace yourself during the exam, but there's something else that you can do once you've actually finished reading the exam. So let's say you finish reading through the exam in 10 minutes. Rather than just then sitting there for 5 minutes waiting for writing time to start, is there something really useful that you can be doing? Any ideas?
It's been a while since I did an exam. I can't remember.
No idea?
No.
Yeah, well, often students do just sit there and just look around and just wait to start writing. But a really useful thing that you can be doing is start answering the questions in your mind. Obviously, you can't write anything, you can't mark the paper, but you can start just slowly working your way through the first multiple choice questions and answering them. And that way, once writing time starts, then you're off and running. You've already worked out the answers to the first 10 to 20 questions, depending on how quickly you go through them. So that gives you a really good head start going into the exam, particularly exams like this one, which is quite intense. You have to be working at it pretty hard for the entire 90 minutes of writing time. All right, so keep that in mind during reading time, really important. Okay, weighting 30%. The exam is at Mel Poly Preston, our Preston campus, Building F, room 145, 146. Don't forget to bring your calculator or your student ID card. Also bring, of course, your pens, pencils, and a clear water bottle. All right, Preston campus map. For those students who've never been to the Preston campus, here's the main road here, St. George's Road. This is the student car park for those students that are driving. And then right next to the student car park, you'll see this fairly new building, Building F. So that's where you're headed to for all your exams. I believe all the exams are being held in Building F. Okay, for anyone that's taking the train, South Nerang line, you can get off at Bell Station or Preston Station, and it's about a 10-minute walk from either of those stations. Okay, and of course there is, I believe there's a tram that goes up St. George's Road. Okay, so that's the Preston campus. Let's look at the exam structure. So there's 3 sections. This is section A, multiple choice questions. Each question's worth 1 mark, and there are 53 questions. Okay, very important for this section to be revising your tutorial questions. Many of those questions will come up again. Some will be the same, some will be slightly different, slightly reworded, but very important that you revise the tutorial questions. Section B, true or false questions. Each question is worth 1 mark, and there are 48 questions in total. Once again, revise your lectures, revise the tutorial questions. You can see already that this exam, you need to be really well prepared to get through it. Students that are really well prepared, that is students that have done all the work during semester, worked at the tutorial questions, made sure that they're understanding all the material, those students will get through all the exam and they'll do really well. So the final exam has a way of really separating out all the students in terms of marks. Every year there might be 1 or 2 students that will get over 90%. And then there's a whole range of marks right down to about 20%. Okay, so obviously there are those students that are really well prepared, they've studied hard, done all the work, and they can achieve those really high marks. And then you have the other end of the spectrum. But what I want to say is that it's possible. Every year there are students that do really well. So it is possible to get really good marks in this exam. All right, so finally, section C, calculations and short answer questions. These are all worth 2 marks each for a total of 28 marks. There are 14 questions in this section, and this includes 11 calculations. Okay, the lectures that will be covered in the exam are lectures 7 through to 18. So they're all listed here. So starting from lecture 7, the chemistry of life, right through to classification. As I said earlier, make sure you revise your tutorial questions. So hopefully everyone's worked their way through all these questions, even the non-assessed ones. If you haven't, the non-assessed online quizzes are still open. They remain open until after the exams. But hopefully you don't need to be doing those in preparation. Hopefully you've already worked your way through all those tutes. All right, here are the calculations that you need to be familiar with. These will all be provided for you on the last page of the exam, but obviously you need to know which ones to use and how to apply them. So PV equals nRT, STP, standard conditions of temperature and pressure, and of course you'll need to know which of these are constants to apply in the PV equals nRT formula. Know how to calculate pH. Okay, our molarity formula, N equals C times V, our molar mass formula. Okay, remember our ion product constant. So how to calculate the concentration of hydronium ions or the concentration of hydroxide ions given the other value. Know how to convert from Celsius to Kelvin, and how to calculate weight per volume percentage and volume per volume percentage. Okay, so make sure you revisit these calculations so that you feel comfortable with them. All right, practice drawing key organelles that are found in cells and know how to label them. So let's just look at one example. All right, and I'm going to draw a chloroplast. Okay, so how would we label this outer layer?
Cell wall.
No, so this is just the chloroplast, not a cell. Just the organelle called a chloroplast. So this would be the outer membrane. Outer membrane. I won't write the whole word. And then you would draw the inner membrane and label that. Inner membrane. Does anyone remember any other part of the chloroplast?
The vacuole?
No. 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 So this is not the cell that I'm drawing. This is just the chloroplast itself. So these internal membranes are called thylakoids. I know it's been a while since we did this lecture. Thylakoids. This was from the functioning cells lecture. So thylakoid membranes, and then these stacks of thylakoid membranes are called grana. Grana. And then this fluid interior is called the stroma. Okay, so that's how you would draw and label a chloroplast. Can you think of any other organelles, cell organelles, that you might be asked to draw?
Golgi.
Golgi, yes, Golgi, that's a possibility. Golgi apparatus, yes. Any other examples?
No. Nucleus, maybe?
Nucleus, maybe, yes, nucleus, Golgi. So we've just seen chloroplast. What else? Mitochondria.
Yes.
Mitochondria, that's a possibility. And endoplasmic reticulum is another possibility. Okay, so make sure that you can draw them and label them correctly. Okay. All right, so that's from our functioning cells lecture. These two here are from the lecture Movement Across Membranes. So make sure that you can distinguish simple diffusion from facilitated transport. Also, make sure that you know the difference between channels and carriers. So remember, two different types of membrane transport proteins. Practice exam. So for first semester, first year, I do give you a practice exam. That'll be up on Moodle in the next day or two. I'll let you know when I post that. Keep in mind that this is a very, very long practice exam, so don't expect to be able to do it within an hour and a half. It might be useful for you to do some revision, then attempt the practice exam, and then use that to guide your further studies in preparation for the exam. So, does anyone have any questions?
No, that's pretty.
No?
Pretty soft. Yeah.
Is that helpful?
Yeah, definitely. Yeah, I think it's
Yes?
pretty helpful. Yeah.
Yep. And one more question. How long do you think you need to spend studying for each exam? And obviously this is, you know, different for different people, but just as a rough guide, how long do you think you should study for one exam in order to get a really good mark?
Probably a good week.
A week?
Yeah.
Yeah, possibly. You know, obviously it depends on how much work you've done during semester. I always think that 3 full days is a good, you know, a good rough estimate. 3 full days of study per exam, and this is assuming that all the understanding is already in place. So that 3 days of study will include obviously revising the material, maybe preparing additional summaries that you didn't have time to do during semester, summarising your summaries, which is always a good technique, to take, you know, a summary that might cover, you know, 3 pages for a particular lecture and trying to get that down to 1 page. And then, of course, looking at those key points on that 1 page, you learn to recall all the information that was on that 3 pages. I think that's quite a good technique for a lot of students. But obviously, you know, it depends on how you learn best. Some students prefer a visual guide, something visual, and that enables them to study much better rather than just reading the words and so on. But I think that's a good one to aim for, about 3 days of full study per exam. So that's it from me.
Thanks a lot.
Thank you, everyone.
Yep. And Pracs due on Friday?
Yes, PRAC report's due on Friday, correct?
Okay.
Yes, Friday at 5 p.m.
Yep. Okay.
So I'm sure I'll have lots of marking to do from Friday onwards, even though I already have a lot of marking to do from other subjects.
Yep.
All right, it's been terrific. Thank you very much, everyone,
Yep.
for participating, for being online, and just thank you very much for all your interest and your great attention.
An interesting subject, I think.
Oh, terrific. Good to hear. That's great to hear. Thanks, everyone, and I'll see. Yes, go on. Yep.
It's unrelated. I wanted you to kind of finish before I went there.
Yes.
Thinking of chemistry and the makeup of different elements, are the protons that make up the different elements or the subatomic particles, are they the same across the different elements, or do they differ per element?
As in the electrons and protons and neutrons?
Is a proton across carbon and hydrogen and oxygen?
Yes,
Yeah?
it is. A proton is a proton. Yeah, absolutely. It's the same.
Subatomic particles that make the difference that we notice. Correct.
And even though we don't study any nuclear chemistry in living systems, if you read up about a little bit about nuclear chemistry, you'll see that you'll see how certain elements can actually lose certain subatomic particles, and they become completely different elements.
Fascinating.
Which is fascinating.
Very interesting.
Yeah. Yep. So when we talk about nuclear decay, you'll have one element that suddenly becomes another element. So it's simply the number of those subatomic particles that actually, contributes to the identity of the element, which means that they're all the same, regardless of which element they actually come from.
Yeah, it's where you start to get into like nuclear fusion.
Yeah, that's right. Yeah, it's a shame that we don't learn any in living systems because it's a very, very interesting topic, nuclear chemistry, but it's just not part of the curriculum for this subject.
Thank you.
Yeah, you're welcome. You're welcome. Well, have a great week, everyone. I look forward to all your prac reports, and I will see you on exam day.
Will you be there?
I will be there for reading time.
Thank you.
Yeah. Okay.
Well, thank you, Silvana. Yeah.
Yes, all the best, everyone.
Thanks, Silvana. See you then.
Thank you. See you then. Okay, bye-bye.